In this video, we are going to be looking at chromosomes and the fact that chromosomes have to condense before cell division can occur. So first we will remember what chromosomes are, and we'll look at defining chromatin. We will define nucleosome, and finally we will describe how the chromosome's appearance changes after DNA replication. So remember that eukaryotic cells, which are things like fungi and plants and animals, that they divide by mitosis. So they don't do binary fission like what we just looked at with our prokaryotic organisms. Instead, it's actually mitosis, which is very similar in terms of what it looks like is going on, but it's much more complex. So your book uses the analogy in discussing chromosomes, which are the DNA in the cell. They compare it to a ball of yarn, which I think is a pretty good analogy because a cell, when it is not in the process of dividing, the DNA is just kind of loosely packed into this sort of a more or less central location of the cell. And you can look at a pile of loose yarn, and that's kind of how the DNA is. It's just very disorganized. And so in terms of trying to copy it, that would be a very difficult thing. On the right, you see a picture of DNA that has condensed into visible chromosomes. So now it's a lot more organized. It's going to be a lot easier to move around and work with. So again, DNA is loose in the cell when it's not dividing, and the phases of the cell cycle that occur when the cell is not dividing are going to be DNA replication. So the cell is not dividing when it's making a copy of itself, and then protein synthesis, making the proteins that are a part of the associated structure of the DNA. That all happens when the cell is in one of its resting phases. DNA condenses when the cell is dividing, and that is when the chromosomes actually take on the form of chromatids, and we'll talk about those later in this video. And as you can see, if you can imagine dividing that genetic information now, it's going to be a lot easier than when it was just loosely packed into the middle of the cell. So in looking at the structure of a chromosome, first of all, we have just the naked DNA. So think about our DNA structure that we talked about in the last video. And it wraps histone proteins. There are 8 histone proteins in each of those bunch so that the DNA kind of looks like it's on beads. And all of that is wrapped into what is called a nucleosome. So the DNA wrapped around those 8 histones forms what we call a nucleosome. Those cluster together into chromatin, and they are held together by what are called scaffold proteins. So in order to keep that structural arrangement, those scaffold proteins form sort of a backbone for keeping that chromatin very tightly packed. So after DNA has been replicated, those chromosomes, as we saw in the original picture, they form. So now they're condensed inside the cell. And again, they take up a whole lot less space in that form, and they're a lot easier to move than when it's just an unwound, tangled mess. So those chromosomes, let me go back for a sec. Previous. So one of the things that I want to point out is those chromosomes, when they are replicated, there are now 2 of them. So those chromatids, the chromatid pair is called sister chromatids. They are held together by the centromere here in the central location. So now we are kind of set up for cell division to occur. So the relationship between chromosomes and chromatin, remember that a chromosome is composed of chromatin, which is basically the DNA and its associated proteins. So in this video, we defined chromosomes and chromatin, we defined the nucleosome, and we described how the chromosome's appearance changes after DNA replication. Remember, it starts out, it's very loosely organized, and then it condenses and turns into a structure that's going to be a lot more easy to move around the cell and to manage.